This is a leaf from a northern red oak. And this is a leaf from the same tree. Both bear the same DNA, yet they look radically different, and they function differently too. This leaf is a sunleaf. It lives in the top of a canopy or around the edges of a tree crown that catch a lot of sunlight. And this leaf is lighter in color, thicker, smaller, and more lobed. We know that this shape allows temperature regulation of the leaf while it is being heated by all of this direct sunlight that it intercepts. We also know that the leaf is able to exchange water and CO2 with the atmosphere at a relatively high rate, and it photosynthesizes more quickly. This, on the other hand, is a shade leaf. It comes from the inside of the tree crown, where a lot of the sunlight is blocked by the leaves that surround it. It has a darker green color, it is thinner, it is less lobed, and it has a much larger surface, which helps to intercept as much sunlight as possible, given the shaded circumstances. It, on the other hand, photosynthesizes more slowly than a sunleaf. As you can tell, we have a pretty good functional explanation of why these leaves look the way they look. They are optimized for photosynthesizing in different light environments. But the developmental part of that why question has never really been answered. Given that these leaves are genetically identical, which information or processes cause them to differentiate, and when during development did these two leaf shapes first become distinct? Hi, my name is Kristel Schoonderwoerd, and I'm a postdoctoral researcher in the Department of Ecology and Evolutionary Biology. My research interests are centered around resting buds in temperate trees, as well as leaf development and leaf aging in deciduous forests. And ever since I first started collecting seasonal leaf data, this question has been on my mind. Most textbooks would have you believe that the functional explanation and developmental explanation for sun and shade leaves are the same. Sun leaves are sun leaves because they develop in a sunny environment, and shade leaves are shade leaves because they develop in a shaded spot. Yet, in this part of the world where a lot of our forest canopies are deciduous, there really aren't any marked differences in light environment for up to 6 or 7 months of the year when the trees bear no leaves. And that period includes the early spring, the time when most leaf expansion happens. In other words, it is unlikely that differences in the light environment are directly driving differences in leaf shape and function at the moment that leaves are actively expanding. This, to my mind, can mean one of three things. 1. Sun and shade leaves diverge in shape at a very late developmental stage when the canopy is almost fully formed and the leaves have already completed most of their expansion. Alternatively, sun and shade leaves diverge in shape at an early developmental stage. Foliage leaves are actually first initiated within the confines of a resting bud. This happens during the summer or fall prior to tree dormancy and, importantly, prior to the loss of leaves from deciduous trees. So theoretically it's possible for this year's leaf development to be influenced by last year's light environment. And for the third option, we need to consider that the bud is really a sequence of leaves, all of which are slightly different ages. The earliest leaves are more developed than the later leaves during dormancy in the bud, and possibly these first leaves to emerge from the resting bud are neither sun nor shade leaves, and it is the younger leaves that are initiated later and also expand slightly later in which the shape divergence occurs. To find out which of these three scenarios is closest to reality, I have designed a field experimental setup using forest edges at the Yukon forests. To me, what is cool about this project idea is that it integrates ecology and developmental biology, which are two fields that don't meet very often. Together, however, they can teach us how the environmental characteristics of this summer around the time when the resting bud set is taking place can influence leaf function and photosynthesis in the next growing season. This approach could potentially reveal lags between functional responses and the environmental conditions that cause them. This study directly addresses the limits that development and anatomy impose on photosynthetic function, and ultimately on tree growth and carbon sequestration. This experiment has 3 parts. First, I'd like to install data loggers with light sensors on branches near buds in the late summer and fall, and half of those will be on branches receiving highlight and the other half will be in shaded spots. So then I'd like to track how over time, how the 2 light environments converge as leaves are lost in the fall, and how quickly the environments diverge again in the spring. Secondly, I'd like to manipulate the light environment of some branches and buds. By introducing and removing shade cloth, I can make changes to the light environment at different times in the season, in addition to the change that is naturally occurring. Third, in parallel to the light measurements and manipulations, I will be collecting samples for microscopy investigation. These include resting buds in the fall and spring, and also expanding leaves in the spring. The best trait, I think, to track will be the development of the mesoville. This is a layer in the leaf that includes most of the photosynthetic cells. And in shade leaves, it is up and thin with 1 cell layer, whereas in sun leaves, it is thick with multiple chloroplast-filled cell layers. Using traditional tissue sectioning and microscopy, I should be able to track cell layer number and cell size across the samples at different developmental stages and work out when the 2 leaf categories first start to diverge. Ideally, I'd like to conduct this experiment in 2 species to specifically address the 3rd hypothesis regarding a different response to light in leaves that expand early in the season and leaves that develop late in the season. The first species would be black birch, which is very abundant in Connecticut and has both early and late leaves. And the second species would be pignut hickory or shagbark hickory, depending on availability. And this species only ever expands leaves early in the season. It doesn't have any late leaves. So the aim here is to examine if these different seasonal leaf growth patterns in different species affect how leaf development is influenced by the light environment. Developmental studies in forested ecosystems are important for truly understanding when during the season CO2 uptake from the atmosphere is occurring and what factors influence how effectively that happens. And when it comes to the influence of light environment on photosynthesis specifically, this will be an important environmental factor to consider as we are introducing trees to human-created environments with entirely new light regimes. Think of forested ecosystems becoming increasingly fragmented and of tree management in increasingly urban landscapes. I'd love for developmental biology to become a tool in predicting and understanding how our tree species photosynthesize and fix carbon in the Anthropocene, and for how trees can be most effectively used to improve our environment as we face climate change. Thank you so much for listening.